Okay, so let's get started. Good morning. And as far as I understand, this lecture also being recorded on video. So for those of you who will be watching it later, also good afternoon, good evening, and good night, I guess. Anyway, so today we will be starting our discussion on nuclear receptor signaling. Can you hear me well? Or no? Okay, so let's see. Let's see how this might work. What about now? Better? Okay. All right. So we have several goals to accomplish today in these 2 hours of the basic cell signaling lecture. And so it's very, very different from the lecture you just heard before. So very little chemistry, although there will be some, and a lot more molecular biology in at least first hour. Second hour will be more probably descriptive. Okay, and basically what we need to do is to talk about cell signal transduction as shown on example of the pathways that are activating nuclear hormone receptors. It's a specific class of proteins which is regulating gene expression in cells and tissues in response to small organic molecules called hormones. And so idea is that you should be able, by the end of this lecture, to name and draw nuclear hormone receptor structures, at least linear maps, and also nuclear hormones, at least have a general idea how hormones that bind to nuclear hormone receptors look like and be able to recognize them. I don't want you to draw all of the details of the structure of the estrogen with all stereochemistry and connectivity involved, but I want to make sure that whenever you see a structure of the estrogen, let's say somewhere painted on a wall, you can say, wait, okay, great, this is what I know, this is the estrogen. Or if somebody in a bus or in a, I don't know, on a metro asks you, how does hormone look like? So you should be able to, to take a pencil and pen and explain it to them. Or if your grandma is asking, how nuclear hormone receptor works, you should be able to answer to her. So this is what happens. You get a binding event, and then you get a translocation of the nucleus and activation of gene expression. Anyway, so this is the idea, basic objective of this lecture. And so in terms of the principles of signal transfer, we will talk about how small molecule hormones, by binding to nuclear hormone receptors, actually turn on or off certain groups of genes. And then finally, we will have to delve a little bit into the mechanism of action of the nuclear hormone receptors and ask questions. Why do they do things that they are doing in the nuclei? How do they do it? And what is the difference between mode of action of different nuclear hormone receptors? Okay, so let's start with a history of nuclear hormone receptor signaling. And you can see that history dates to exactly the same year that USC School of Pharmacy was founded, 1905. So it's a long, long time ago, more than 100 years ago. And yet, no, basically, USC School of Pharmacy doesn't have any connection with this date. But same year that USC School of Pharmacy was founded, the Ernest Starling was coining word hormone, and it's from Greek horm, which means the one that actually acts upon something or has some function, some property. And so that the message that is sent by the hormone basically is affecting cells either in the same organ or in a different organs in the body. So essentially, body uses hormones to send a message to other organs and to alter their function in some particular predictable way. Oftentimes, hormones have been produced in one organ and then been carried out to others, although not always. Okay, so now, then about 21 years had passed, and Kendall and Reichstein finally were able to isolate and determine chemical structure of two hormones. One of them was called cortisone, and the other one was called thyroxine. And so that was because of the successes of chemistry as a discipline, and finally because of the ability of chemists to determine structure of the compounds of the small molecule. And then a few more years, structure of estrogen was determined, and interestingly enough, it is a fairly complex molecule. Nonetheless, both isolation and structural determination happened at that year. So when the organic chemistry was just basically making its first few steps. And then another few decades have passed. So you can see 1958, and finally estrogen receptor isolation was performed. And so you can see the difference between complexity of the protein and complexity of the small molecule is such that it requires several decades to elucidate a structure of the protein after the structure of the small molecule that binds to it has become known. And then finally, only in 1980s, because of the successes of molecular cloning and molecular and cell biology, finally cloning was done on most of the nuclear hormone receptors, including estrogen receptor, glucocorticoid receptor, and thyroid hormone receptor. And then only in 2004, almost 100 years after word hormone was coined, the people who did cloning on these receptors won the Albert Lasker Award, which is oftentimes in medicine considered to be equivalent to the Nobel Prize. So there is Nobel Prize in medicine, but there is also Lasker Award, which is more on the side of the basic research in medicine. Okay, so that's the history of the nuclear hormone receptors and the field that evolved as a result. All right, so what is the nuclear hormone receptors and what do they do? First of all, they are proteins that interact with small organic molecules, and small organic molecules can diffuse freely through the plasma membrane inside the cell. So nuclear hormone receptors are located inside the cell. They are not generally found on the membrane. That's the important first point. Second is that when this binding event happens between hormone and hormone receptor, actually then the receptors become activated. And activated receptors then go into the nucleus, or as biologists call translocate into the nucleus, and bind to the specific stretches of the DNA in the promoters of the genes, which are called HREs, hormone response elements. So hormone response elements are basically DNA sequences that are unique to that particular hormone receptor, and the receptor is able to recognize them and bind with very high affinity. Okay, so these are just examples of several of those hormones. Androgens and estrogen receptor are probably two of the most often mentioned and probably also most widely studied. In addition, there are others, like, for example, vitamin D receptor, thyroid hormone receptor, and thyroid hormone. And then there is also retinoic acid receptor, and so on. Yes?
So are the nuclear hormone receptors, if the nuclear hormone receptors are not on the membrane, are they free-floating in the cytosol?
Well, to tell you honestly, no protein really free-floating in the cytosol, but yes, they are located in the cytosol. So they follow their particular compartments even there in many cases. But yes, you're right, they are pretty much free to move to some extent in the cytosol, and then once they activate, they go into the nucleus. We'll talk about this in a moment. Okay. So one important thing that you should remember from this particular slide, that all nuclear hormone receptors basically have the same general function, when they are bound by hormones, they act as transcription factors. So all of them are functionally related. So they are inside the cell, and when they are bound with the hormone, they act as transcription factors. So as a result, they will be either activating or sometimes repressing transcription of certain genes. So the formation of the messenger RNA from the genetic code, whatever it is in the DNA for the particular gene. Okay, so how do they look like? So now, so that the structures of nuclear hormone receptor have been elucidated both biochemical means and in many cases also by X-ray crystallography. So we can look a little bit more into detail what do they have. So first of all, if you look at the protein structure, you probably want to start somewhere, right? So if you say, well, nuclear hormone receptors such as estrogen receptor has this many amino acids, so you probably will start looking from the N-terminus. This is where the terminal amino group of the large protein sequence starts. And you kind of go from N to the C terminus of that large, peptide sequence or the protein sequence. And so at the N-terminus, you have so-called activator function 1. And that is basically the sequence of amino acids that hormone receptors use to interact with other parts of the transcriptional machinery. We'll talk about this as well. So that's the N-terminal region, or N-terminal regulatory domain. Then a second component, which is also very important in the structure of the nuclear hormone receptor, is DNA binding domain. They must bind to DNA in order to function as transcription factors, so that clearly that region is responsible for interacting with their unique response elements. So DNA binding domain, or DBD, is interacting with HRE, with hormone response elements on DNA. Basically, they are structurally similar to the class of proteins which are called zinc fingers. And so that basically some of them contain zinc atoms in the structure in a way so that it specifically positions certain parts of that protein toward DNA, toward the double-strand DNA. And then there is a hinge region which connects essentially DNA binding domain with other part of the protein, which is called ligand binding domain. And this is very random region oftentimes that just serves a structural purpose. It will provide some connectivity between these several domains. And then finally, ligand binding domain. And this is very important domain because this is what basically interacts with hormones. So that particular region, that particular domain of the protein is responsible for interacting with hormone and causing certain structural change upon this interaction with hormone so that the protein can then translocate into the nucleus. Okay. And then finally, there is a C-terminal domain, and that one serves various purposes. In some nuclear hormone receptors, it's actually responsible for recruiting certain classes of the proteins called coactivators and co-repressors, and we'll talk about it again later on. Also, in other situations, it is responsible for dimerization of this or the other transcription factors. So essentially, when they're in the nuclei, C-terminal domains might touch each other and help to form dimer from two copies of identical nuclear hormone receptor. And in others, it doesn't serve any particular purpose. It just basically there maybe because of the evolution, or maybe it maintains the overall solubility of the protein. So these are the domains. So starting with A and B and all the way to the F. So these are the classes. So essentially five domains. Each of them serves particular purpose. And this is the way they look like. Yeah.
So does the AB domain, the N-terminal, it does the regulating of the receptor?
What it does basically, it is causing it to recruit, or it is attracting certain proteins from other parts of the transcriptional machinery. So regulation of the receptor actually happens through the ligand binding domain. So this is where a hormone binds. So in this case, you can see ligand binding domain of the androgen receptor, and with the ligand, with the hormone bound, and so that one is then causing changes in certain parts of the structure of that domain, and then it propagates through the protein, causes it to be able to be imported into the nucleus. And so this is essentially what the linear map of the nuclear hormone receptor in general look like. And so you start with the N terminus, and you go all the way to the C terminus. And you can see the ligand binding domain by far is the largest. And it makes sense why it is the largest, because in order to bind tightly to a hydrophobic small molecule, which hormones, most of them are, you need to have a lot of protein structure for specificity. And this is what ligand binding domain to different hormone receptors provide. And also DNA binding domain has to be also highly structured, because what it does, it interacts with the double-strand DNA in the nucleus. And so it has to provide very high affinity of interaction of that protein with DNA. For different types of nuclear hormone receptors, that domain is slightly different. But nonetheless, because DNA itself is, even in a nucleus, is a B form, right? So it's fairly similar in shape, despite the fact that maybe nucleobases here will be slightly different for different nuclear hormone receptor. Because of that, so these regions in a DNA binding domain will be relatively well conserved. Okay, so now, how do you classify nuclear hormone receptors? So there are a number of different nomenclatures that people came up with just to make things difficult, I guess. But there is one that's simple. So you can call type 1, which will be ones that will bind to the steroid hormones, and the type 2, which binds non-steroid hormones. So based on these two types, you can then name several hormone receptors like androgen, glucocorticoid receptor, or estrogen receptor, and so on. And so this is easiest classification and simplest. There is also more complicated classification, which I don't really want you to memorize, because this is a nightmare, and I don't remember everything when I look at this. But the reason why this more extensive classification came about is that there was a need to classify and place into particular groups newly discovered proteins that looked like nuclear hormone receptors, but in some cases they didn't bind any hormones. And so then they were usually called like. So like there is an estrogen receptor, nuclear hormone receptor. That's the protein that binds estrogen. But there is also estrogen-related receptor that doesn't bind estrogen, but it binds to the same region in DNA in the nucleus that estrogen receptor does. And so by that similarity, you have to classify somehow these types of receptors. So they made classification by types of classes. And you can see the class 1, you have all of these receptors that basically form dimers, homodimers or heterodimers, and respond to the particular ligand. And you can see in some cases you have receptors that don't even bind any known ligand. It doesn't mean that it doesn't exist, but at this point it hasn't been discovered probably. And then you have another class, class 2, which then again has receptors that form either homodimers or heterodimers, so basically two identical or two dissimilar proteins bind next to each other. And also depending what kind of binding sequence they bind to. So this classification also takes this into consideration. And so if you look at this classification, there will be basically 6 main classes, and then there is also this so-called class 0, which makes it 7. And as a result of this, all of the type 1 receptors that bind steroids, you can see they will fall into this so-called class 3 of the newer classification. Yeah, I just said that basically nuclear hormone receptors that bind steroids will be falling into the class 3 of this new classification. Sometimes they also called subfamily. So class or subfamily is essentially identical in this terms of in this nomenclature. Okay, so that's basically how they are categorized and classified. And then whenever new protein that looks like it is acting as a nuclear hormone receptor, or it binds to the response element of the one of the known nuclear hormone receptors, they could be, placed into these subfamilies or classes, and so making life, at least for molecular biologists, easier. And for us, it's complicated because so many things to see. Anyway, okay. Now, hormones, small organic molecules that bind to nuclear hormone receptors. And as you can see in this slide, you don't have to remember, as I said, structure of every hormone, okay? And I deliberately put these structures without any notion of stereochemistry because this would make things even more complicated. For example, this metal group should be ideally pointing up. So it should be a wedge and so on. But just in this case, I want you to recognize steroids from non-steroids. And it's very, very easy to basically recognize steroid from and be able to say that this is one of the steroid hormones. So if you look at this basic structure of the cyclopentano-phenanthrene, which has been also saturated, basically hydrogenized version. So it's a hydrogen substituted skeleton of the cyclopentano-phenanthrene. So yeah, so the 3 6-membered rings connected to the 1 5-membered ring. So that's the signature of the steroid. Everything else that is not like that is non-steroid hormone. So for example, this is the dihydroxyvitamin D3, and it doesn't look like anything that is above, right? So it's non-steroid. Triiodothyronine, which is the hormone for the thyroid hormone receptor, doesn't look like a steroid anyway. Yeah. Yeah, this is the basic structure of the steroid. So cyclopentano-phenanthrene, which has partially hydrogenated skeleton. So you can see there could be some aromatic ring in here, like the estrogen or estradiol, and could be something like this, which is in cortisol. So this is alpha-beta unsaturated ketone, and so on. But overall is the same basic structure. And then there are other classes of hormones which have their own name, and these are prostaglandins. So the prostaglandins are hormones that are secreted by the prostate gland, and that's why the name. And you can see in this case also they have very well recognizable general structure. You have cyclopentanone and this unsaturated hydrocarbon chain, that kind of two of them side by side that propagate, that come about in space. So these are the sort of signature structure of the prostaglandin. And then retinoic acid, trans and cis, also has very, very well recognizable structure. It's a 6-membered ring with several metal groups, and then attached to this single polyunsaturated chain. And so it could be cis or trans, and all of them will basically be resembling this kind of structure. Okay, that's about it, what I would like you to know about the hormones. Now, just to reiterate, small organic molecules, typically hydrophobic, because there is a lot of hydrocarbon chains attached to them, and that in part helps them to traverse the plasma membrane, because they can embed into the membrane and kind of go right through, and so. So, and also, of course, they bind to the nuclear hormone receptors. And their main site of action is nucleus, because this is where the activated nuclear hormone receptors will normally reside, right? And now, what is also important, especially for pharmacy profession, that yes, they could be either brought exogenously or synthesized in vivo. And in many instances, certain exogenous hormones could supplement what is being synthesized in vivo. And this way, you can relatively easily modulate function of different organs or treat diseases. Like I will show you an example of selective estrogen receptor modulators, right, that used in the treatment of breast cancer. Okay. Let's now compare linear maps of several hormone receptors and see what is similar and what is dissimilar in their structures, right? And the way we can do this, we can actually align all of them in such a direction so that the N-terminus on the left, C-terminus is on the right, and all of their DNA binding domains are pretty much in register. So all of them align in the same sequence, in a similar sequence. And then you can ask question, okay, what are the domains that are most conserved, most similar in all of the nuclear hormone receptors? Right, DNA binding domain. And it makes sense because all of them will be binding to the stretches of DNA which are about 5 or 6 base pairs in length. All of them will have to bind with high affinity, although the sequences may be slightly different, but the range of sequences might be somewhat similar for the similar type of receptors.
Oh, so do they bind to the same sequence or different sequences?
No, all of them will be binding to slightly different sequences, but the number of the bases on DNA will be about the same, 6. Okay. Now, ligand binding domains, on the other hand, are, again, probably next most conserved. You can see there is a similarity between 15 to 57% in amino acid sequence in most of the ligand binding domains. And the reason for this is, again, because hormones, mostly molecules that have similar shape, slightly different maybe hydrophobicity, and so that the molecular recognition principle that nature uses to bind to hormones is about conserved to some extent. So that's why ligand binding domains, all of them will be about the same length and have moderate sequence homology. Okay, now activator function, on the other hand, or N-terminal region, is very, very different for many of these nuclear hormone receptors. And the reason for this is that oftentimes they recruit different proteins in the nuclei, and oftentimes they could bind to other proteins also in the nuclei. And so in order to accomplish that function, you need very, very different the N-terminal domains. And this is what happens. Essentially, there is a 0% similarity in amino acid sequence between different nuclear hormone receptors. And worse yet, you can see that these regions are different in length. In some cases, it could be hundreds of amino acids. In other cases, it could be maybe 10 to 15. Okay, and then C-terminal domain, again, is highly variable. In some receptors, it's long. In others, it is short. And in some situations, it doesn't seem to exist at all. At least only ligand binding domain is ending with the C-terminus, and that's it. And then there's a hinge region. And hinge region, again, people don't know much about its structure because in every case it's slightly different. And depending how you crystallize nuclear hormone receptor, it may assume different conformation. But in many cases, hinge region sort of in between DNA binding and ligand binding domains. So it's moderately conserved as well. Okay, now, where do they bind nuclear hormone receptors once they enter into the nuclei? Well, they will be binding to the hormone response element, and for each case, this particular sequence will be slightly different, but it will be specific for that particular hormone. In addition, depending what type of nuclear hormone receptor is in the nucleus, it will be binding to its response element either as a dimer of itself, so basically two copies of the receptor will sit down on DNA, so to speak, right next to each other. And as a result of this, the DNA sequence, you can imagine that it will be having similar base pair in forward and in reverse direction from 5' to 3' side. The reason for this is if one copy of the receptor binds right here, the second copy will have to be inverted 180 degrees and bound right there. So that's why this sequence, if you go from the 5' direction to 3' direction, will be the same as this sequence that's going from 5' to 3, from 5' to 3' direction. And so that creates sort of palindromic, what's called an inverted palindrome, because the sequences from one and the other direction are the same. So the reason it is that two identical copies of the nuclear hormone receptor will be binding to that sequence. And because of these two identical copies, their DNA binding domains will be same, but they will be rotated 180 degrees, right? And probably better if I just illustrate it. Okay, let's imagine you have two copies of the nuclear hormone receptor, right? And binding to the DNA sequence. What will happen? So one of them will be rotated 180 degrees. The other one will be binding in a direct sequence. And if they bind like this, so you can imagine that they will be now one of them will be occupying this particular subsite, and the other one will be occupying this particular subsite. And because they will be, when they are bound, they will be essentially a mirror image of each other. These sides will be sort of also like a mirror image of each other, but it's only through the sequence from 5' to 3'. You can see on this example. And these three base pairs could be anything. So there is this symbol N means that it could be any base, any nuclear base out of 4. But this direction, AGAACA, is the same as this, AGAACA, right? And so this is because two of the copies of this receptor will be binding simultaneously to the response element. Okay. Yeah? Yes, they interact with each other, actually, and they what's called the homodimerize. So there is a specific region that essentially responsible for the formation of the dimer, and that's on the level of just protein touching another protein. Same story with the estrogen receptor. So estrogen receptor binds to its response element, again, as two identical copies. So they will be now recognizing this particular sequence. You can see it's a little bit different from the glucocorticoid receptor sequence, but it's still going to be inverted palindrome. So from 5' to 3', it's going to be this, AGGTCA. And then again, on the other side, 3 bases away, from the opposite strand, again, 5' to 3' will be AGGTCA. And so that's again because once you have two copies bound and one of them has to rotate 180 degrees, so that's the way how you can accommodate them on DNA. Okay, so this is how the how binding of the nuclear hormone receptors happens, and not all of them will be binding as two identical copies of themselves. They may actually have another friend that will be binding with them, and this is a retinoic acid receptor. And so RXR, for example, binds together with the vitamin D3 receptor and forming this so-called heterodimer. Now, two different proteins bind next to each other, but they will be also interacting with each other through some kind of fold. But as a result of this, the response element will no longer be mirror image of itself and no longer will be inverted palindrome. But it's going to be just direct site. And this particular portion, let's say, will be responsible for binding of the retinoic acid X receptor. And this one might be responsible for vitamin D3 receptor. And so now for the heterodimer, basically you don't have to have inverted palindrome. And the same story is same with the thyroid hormone receptor and retinoic acid A receptor. Okay, now, how do we prove that action of the hormones and nuclear hormone receptors, when they are binding to hormones, actually go into the nucleus?
Sorry, just quickly on that last one. So is it that you have one part of it, because it's like those zinc fingers, you have like one finger that's binding upstream and then one that's binding downstream?
Yeah. Well, essentially, these are domains, DNA binding domains, that are very similar by structure to zinc fingers. Yeah. Every case, be it heterodimer or homodimer, it's still zinc finger type. Okay, so now, upon binding of hormones, nuclear hormone receptors translocate into the nucleus. And how do you prove it? Well, the easiest way to do it is to stain them with something, or basically to make a fusion protein with something that you can see in a cell. And one way to do it is to use beta-galactosidase as a sort of marker that you can visualize later on inside the cell. And you can then do two experiments. So you can take just beta-galactosidase, which is your just marker, and put it on cells in a situation where you have your hormone present. In this case, this is for glucocorticoid receptor. It's a dexamethasone as one of the compounds that bind, that sort of activators for the glucocorticoid receptor. And so if you just take beta-galactosidase and you add hormone or don't add hormone, situation is exactly the same. It doesn't have any ability to bind it, right? And so it's going to reside in the cytoplasm regardless whether you add hormone or not. Now you fuse beta-galactosidase with the full-length glucocorticoid receptor. So you basically made a chimera of two proteins, right? And so once you've done that and repeat the same experiment, the situation is very, very different. You can see in the absence of the hormone, all protein is residing in the cytoplasm. And once you add hormone, the protein is immediately localizing in the nucleus. So again, the glucocorticoid receptor causes the entire fusion protein to translocate into the nucleus upon just activation, upon binding with the hormone. Now, you can ask, oh, maybe there is something special about DNA binding domain, or maybe hinge region of the glucocorticoid receptors, and they actually force whole construct to go into the nucleus. Well, you can chop off these domains. You can just take ligand binding domain, something that just binds your hormone and nothing else. And you can fuse it again to your beta-galactosidase and use it as a marker. And so you can see again that situation is exactly the same. Upon addition of hormone, it translocates, and if you don't have a hormone, it doesn't.
I would imagine it would be, wouldn't it be the DNA domain that translocates to the nucleus?
So now, DNA binding domain helps receptor to bind to the right place in the nucleus. But ligand binding domain is what forces it to go into the nucleus, generally. And so this is the proof. So if you just take ligand binding domain, this very small piece of the entire glucocorticoid receptor, and you fuse it to the beta-galactosidase, it still goes into the nucleus. It can't bind to DNA anymore, but it still stays there. So that basically shows that there are two separate processes that are going on. Right.
Does it clarify how the location domain of nuclear receptors are located in the nucleus?
That's correct. Yes. So essentially, if you go more into details, so the binding site for the importin proteins that translocate, help the whole thing to translocate through the nuclear pore, is in the ligand binding domain. Yeah. Okay, now, so with all that, how the general scheme of activation of nuclear hormone receptor would look like. So this is what is being known for several types of the nuclear hormone receptors. This is example just with the glucocorticoid, same one that I showed you the fusion experiment just a second ago. So in the absence of the hormone, it resides in the cytoplasm, and it just not even resides on its own, because there are several inhibitor proteins that are binding to the ligand binding domain of the glucocorticoid receptor. And those are generally so-called heat shock proteins. I think it's 70 and 90 kilodalton, but it doesn't matter. So these are inhibitor proteins. Once hormone comes in and binds to the ligand binding domain, inhibitor proteins are being displaced because ligand binding domain changes its conformation, basically changes its global fold. And it can no longer accommodate inhibitor proteins. And once these inhibitor proteins are actually out of the ligand binding domain, the important proteins will be binding to the similar, basically nearby sites, and will drug it, pull it inside into the nucleus. And once in the nucleus, now, and once it is in a sufficient concentration in the nucleus, it will be now binding to its response element, because DNA binding domain has very, very high affinity toward of the nuclear hormone receptor, has very high affinity toward its cognate site, toward the hormone response element. Usually, in terms of the concentration, you need about nanomole, 1 to 2 nanomole concentration of the protein to achieve 50% saturation of the binding sites. So now DNA binding domain comes in contact with the hormone response element, and now ligand binding domain in this case oftentimes works as a sort of dimerizer, so it helps kind of to structurize this portion of the protein. And as a result of this, once these two ligand binding domains come in contact with each other, activator domains become placed in the right direction, in the right position, and then they begin to function as a molecular glue, basically to recruit other proteins from the general transcriptional machinery, or from the machinery that changes the structure of the chromatin, of the chromosomal DNA right nearby the site. And the reason for this is once you change structure of the chromatin, then you can place more DNA binding proteins that regulate transcription and start transcription right next to that site. And then afterwards you get the transcription to go on. Okay. Now, so I just mentioned the function of the activator domain, this N-terminal region. And so you can ask, what kind of proteins these activator domains interact with? Well, these are so-called co-regulatory proteins. And they belong to two classes. First class is called coactivators. So these are the proteins that are recruited or bound by the nuclear hormone receptors once they are in nuclei and activated by their respective hormones. Or sometimes they call agonist ligands. That's basically the same as the equivalent of the hormone. So that's the ligand that binds, or it's going to be wild-type natural ligand that binds and causes nuclear hormone receptor to function the way it should, to turn on gene expression. Okay, so now these proteins have one important function. So the function is called the histone acetyltransferase. And what it means. So as you know, in eukaryotic cells, DNA in the nucleus is not just in a random state. Yeah, right. It's put on a proteins called histones, which together, 8 of them, form so-called histone octamer. That then comes together with other histone octamers, and it forms what's called nucleosome core particles. And so then the chromatin fibers are comprised of these core particles. So you can see the DNA inside the nucleus is very well organized, structured, and compacted in such a way that it's wound on these histone proteins. And so histone acetyltransferases will basically unwind DNA from the histones. They will acetylate lysines of the histone proteins. And in this way, they will decompact chromatin, decompact nucleosomal core particles, opening DNA for transcription, making it more accessible for the proteins that then govern general transcriptional activation to come in and bind. And then also you need to open up enough space to allow recruitment of the RNA polymerase II, right? Because this is the enzyme that is essentially responsible for the synthesis of the messenger RNA. So you really need to open up this chromatin. And this is what coactivator proteins do. They make this first critical step in decompaction of the chromatin and preparation for transcription. Okay, so this is what I just said, basically. So the weakening association of DNA with the protein. Okay, so there are also proteins that reverse the same process, and they are called corepressors. And in this case, corepressors do exactly opposite function to the coactivator. So they deacetylate histones, strengthen association of DNA with histone proteins, and basically compact chromatin back. And this happens when nuclear hormone receptors are either not present, or they are present, but there is a special situation when they are bound with different small molecules, which are called antagonist ligands. And so when the nuclear hormone receptors are bound with antagonists, oftentimes there are a ligand binding domain and activation domain interact in such a way that they will recruit, instead of coactivator proteins, they will recruit corepressor proteins. And this way you will downregulate gene expression instead of upregulating it. And you can imagine this is a nice way how nature devised basically in a way to tune up gene expression. Yes? Generally speaking, that depends upon the HDAC and HAT. Most of the time, they don't. In some situations, there are associated factors with them, which may. And we'll talk about proteins called P300 and CBP, crab binding protein, in the next hour. And in that case, I can show you what domains of these proteins could actually recruit other parts of the transcriptional machinery. Okay. All right, so let's take a break. So since already it's almost 11 o'clock, we'll take 5 minutes break, and then we'll continue. Right, so let there be the light, and so happened, right? All right, let's continue our discussion of co-regulatory proteins. Just because they translocate into the nucleus and activate gene expression, it doesn't always mean that they have to bind hormones. They have to always bind hormones while in cytosol. And so there are 2 different types of mechanisms by which nuclear hormone receptors are being activated. And the easiest way to remember them by the classification of the hormone receptors themselves. So the receptors which form homodimers, typically they reside in a cytoplasm. And typically they are bound with inhibitory proteins, just like I showed you an example of the glucocorticoid receptor. Okay, so they bound with the inhibitory proteins, normally known as the heat shock proteins, and those will block the localization sequences that could force them to be imported into the nucleus. Now, once hormone comes in, inhibitory proteins are being displaced. Hormone is bound. Now, 2 things may happen afterwards. Oftentimes, ligand binding domain with hormone bound will also force this protein to form homodimer right in the cytosol. And then the homodimer is being imported inside into the nucleus. It doesn't always have to be the case. In fact, in some cases, just the monomer can go inside, bind to the hormone response element, and then the other monomer will bind here as well, next to each other, and then they will dimerize. So it could be one or the other way. So I'll just give you one example where the dimerization happens in the cytoplasm. Timing for that dimerization also subject to a lot of controversy, and sometimes they say it takes minutes, for others say it takes about hour or so to dimerize and then bind to DNA. But whatever the time is, eventually they end up on DNA in a bound state. In bound state. And now, at this point, their activator functions will be recruiting co-activator proteins. And so once co-activator is being recruited, as I said, the DNA will become decompacted because histones will be acetylated. And then DNA decompacted will become binding site for the other parts of the general transcriptional machinery. And then eventually this will result in a recruitment of the RNA polymerase II in the case of the eukaryotic cells. And as a result of this recruitment, you get now elevated level of expression of the gene that has this hormone response element. So it could be tens, it could be hundreds of different genes which are upregulated, turned on basically by the binding of the nuclear hormone receptor in its activated state. So this is one mechanism how the binding with hormone and dimerization happens. There is a second mechanism, and this one is more common for the heterodimeric nuclear hormone receptors, ones that form heterodimers, like this particular thyroid hormone receptor. So in the absence of the hormone, instead of being in the cytoplasm, now this receptor is already heterodimerized and it is in the nucleus, even without hormone. However, it is bound by co-repressor protein. And so co-repressor in this case keeps histones deacetylated, DNA is compacted, and no gene expression takes place. Now, once hormone comes in, the change happens to the ligand binding domain such that co-repressor is being released. And so now the hormone is bound, co-repressor is being released. Instead of that, co-activator comes in and is being recruited to that ligand binding or activator function. And then once co-activator is being recruited, usual things happen, so you get, again, decompaction of DNA, and then other proteins come in, including RNA polymerase II, and you get gene expression. Yep.
So the co-repressor it's not the same thing as HSP, but it does the same thing?
Okay, very interesting. Actually, in a way, yes, but the co-repressor blocks the global activity of this heterodimer for gene expression. HSP, in case of homodimer right here, only prevents nuclear hormone receptor from entering nucleus. But in a way, in a sense, yes, functionally, they sort of act the same way because you get no gene expression, right? Okay. Yes?
So, how is that hormone getting, it has to go through the initial plasma membrane
Yes.
into there, and then why wouldn't heterodimers just normally be the predominant form?
So the hormone, first, to answer your first question, hormone is a small organic molecule, and once it gets through the plasma membrane, it can get into the nucleus very easily because nuclei have pores, which are about 40 angstroms in size. And so if it can get through the plasma membrane, it can get through the nuclear membrane very, very easily. And then in this case, most of the heterodimers of this hormone receptor will be bound to the cognate site inside the nucleus. That's the way how things were set up originally. Then once they have been expressed inside the ER, they've been immediately imported into the nucleus, regardless of whether hormone bound or not. They are binding to their site, and then it's only activator function that works in a different way. This is where the switch happens. Okay. All right. so now we talked about general mechanism of nuclear hormone receptor activation. So let's go to the specific examples and details. You know, devil is always in details, and we will go into one of them, okay, right, into one example of activation of the nuclear hormone receptor. So let's talk about estrogen receptor. Okay, so now the correct name for estrogen, for the hormone, is this. You don't have to memorize it. It's just one example that tells you how many different structural elements it has, right? So, and that's the right configuration and right stereochemistry of this molecule. So again, you can see this is steroid, has this skeleton of the cyclopentane of phenanthrene. One of the ring of the estrogen is aromatic, and actually this is aromatic phenol. And so there is another hydroxyl group, and you can see all of these structural elements, all of the hydrogens, metal, and OH are pointed in a particular direction, and that's very important. So that's the way how natural estrogen looks like. If you reverse or invert at least one of the stereocenters, it's going to be a totally different compound, and it may not even bind to the hormone receptor anymore. And the total number of all of this permutation could be actually quite large. So you can see there are 4 different stereocenters, and you can have one or the other stereo configuration. So 2 to the power of 4, right? So that's what? 16? Okay, so estrogen will be 1 out of 16 different compounds that will have same basic skeletal structure, just different stereochemistry. And you can imagine in some cases you can have even more stereocenters. In some cases, it could be as many as 8. So that's 256, right? And so on. Anyway, all right, so one thing that I wanted to tell you again, just to emphasize, that we will not be talking about proteins that are in a membrane. Oftentimes, there is also so-called G-protein coupled receptor 30, estrogen G-protein coupled receptor. It also binds this molecule, but it doesn't do anything to gene expression. It has completely different functions, so we won't be worrying about it in this lecture. So that GPR30 is another example of the endogenous protein in cells that bind hormones, although they don't serve as the transcription factors. Okay. All right. Now, as I said, devil is in details, and this is one detail. First of all, there are 2 different forms of the estrogen receptor, alpha and beta. And so if you take the linear map of one and the other and superimpose them, you will find out that they are quite actually dissimilar. Both will bind estrogen, same way. Both will activate gene expression in a very similar way. However, they will be different in terms of the response to other estrogen-like molecules. And we will talk about this. This actually is the example of how you can tune up response to one or the other isoform. And interestingly enough, alpha and beta isoform are essentially 2 separate proteins which are encoded by 2 different genes. So it's not something like a splice variant, so you have one gene and then you get messenger RNA. Splicing is different, and you get several different isoforms of the protein to form. No, these are 2 separate, completely different genes. Then the other thing which makes them different, besides obvious difference in length here in C-terminal domain and this N-terminal region, there is also different possibility for phosphorylation of this receptor. And phosphorylation appears to be very important in regulating its activity as a transcription factor in a nucleus. You can see in one case, this region that is phosphorylated may actually prevent estrogen receptor beta, but not alpha, from binding to DNA. Because you put a negative charge right into DNA binding domain that will be supposed to be interacting with DNA. And that's one way how a certain group of proteins, kinases, may actually modulate activity of the estrogen receptor beta, but not alpha because it doesn't have it in there. Okay, so now, yeah.
I'm seeing a lot of familiar names like ERK and AKT and PKA.
Yeah.
So I think aren't those like factors that help grow the body?
So some of them include these factors that activate certain cellular kinases, and AKT is basically one of these proteins that does respond to the growth factors. I put this specifically then if you want to delve why these phosphorylated sites are placed, you can look at the signaling of these proteins, but you don't have to do it for this class, it's just if you're interested. Okay? Yeah?
You said the beta is phosphorylated at a point that...
This point, right. Yeah, that point where there's DNA binding domain.
Oh, that's the DNA.
Yeah, that's its DNA. You can see N-terminus, activator function, DNA binding domain, ligand binding domain, C-terminus. Okay. No. Okay, now, to make things even more difficult, now you can imagine alpha and beta could form either homodimers of themselves, alpha-alpha and beta-beta, but it could be also form alpha and beta. And so now how do you call alpha and beta? Technically it's heterodimer if you use terminology of molecular biologists, right? Because there's two different proteins, all right? But functionally, in response to estrogen, they will be acting the same way as alpha-alpha or beta-beta. So that means they will be binding to the inverted palindromic repeat of the DNA, just like these two would bind. So that's why this word heterodimer, I would highlight it, and I would say it's a structural heterodimer. Essentially, these are two different proteins, but they sort of work the same way in response to estrogen. Yep.
So can we say that homodimers will bind palindromically and heterodimers will bind like...
Yes, you can say that, yeah. And we'll talk about this once we go to other examples. Okay, now, this is how ligand binding domains of alpha-alpha and beta-beta homodimers look like. And estrogen comes right there, you can see in this example in the beta-beta, and I think it binds right here in the alpha-alpha. So by doing crystallography work, structural biologists were able to see how the two different isoforms are being activated. Okay, can you hear me now? Anyway, all right, so how these two isoforms are being activated. This became a basis for development of selective estrogen receptor modulators, if you want to activate one isoform versus the other. Okay, so now what happens when estrogen binds to alpha-alpha or beta-beta form? Actually, something happens, and I said, you see, this is a classical example. This is what I just told you about the activation of the estrogen receptor. So this part, you shouldn't worry about. And the reason why I'm showing you everything is that you can appreciate that the hormones sometimes interact with a number of different proteins in a cell, given the complexity of the pathway. And what we're learning is essentially this small portion where you have activated estrogen receptor as a homodimer, and then it binds to DNA. This is the estrogen response element. And then there is a recruitment of these proteins that I told you. So after histone acetyltransferase, other proteins come in, and then you get activation of gene expression. So, but there are many, many other proteins with which activated estrogen receptor can actually interact in the nucleus. And different cells, different tissues will be responding to activated estrogen receptor in slightly different way, precisely because there is different proteins that might be interacting with them in the nucleus. Okay, now, what happens when estrogen binds to the estrogen receptor? So first of all, some structural changes take place. And so if you take look at this picture, so there is one portion of the ligand binding domain of the estrogen receptor that undergoes structural change or conformational change. And this is so-called helix 12 in green, which in presence of the estrogen basically wraps around binding site. And now it's being kind of converted into the two helices, which are now separated by this little linker region. And that's enough now to change the global shape of that entire domain and cause import of the estrogen receptor then into the nucleus and where it binds subsequently with DNA. Okay, this is probably a little bit better option to show it. So this is helix 12 without estrogen. This is now helix 12 with estrogen. And you can see kind of forming this clump that binds next to the estrogen binding site. And so that little structural repositioning of that helix is the mechanism for the activation of that receptor. So, and question is why people wasted their time to do this and make all of these structures and try to find out what happens to this helix 12 and so on. Well, it appears to be that the aberrant overactivation of the estrogen receptor happens during the early stages of breast cancer. And blockade of this function of the estrogen receptor, especially in the early stages, is important in cancer treatment. However, you don't want to shut down expression of the genes which are on the estrogen receptor control in every part of the body, right? You just want to do this selectively for the tumor. And so in this situation, having designed small molecules that might be able to selectively bind alpha homodimer versus beta-beta or vice versa may be beneficial, because in this way you will be modulating or regulating gene expression only in the diseased tissue and maybe sparing the healthy tissues. And so in order to be able to design these molecules that will look like estrogen but now will be selective, you do need to have some structural information about the ligand binding domain of the estrogen receptor. And so that's why, people spending time and money and effort on this. Okay, now what kind of molecules you can design? Well, actually quite a bit. So medicinal chemists do have a lot of knowledge of how to make design molecules based on a known crystal structure of the receptor. And so they basically have done this quite well. An example of the designed small molecules that look like estrogen or estradiol, but now being more selective towards certain isoforms right here. And so you can see in this case, so if you take estrone, which does look like estrogen except it's a ketone, not an alcohol, so this one will be binding preferentially to the alpha isoform. And raloxifene, this should be N, by the way, which doesn't look like estrone or estrogen at all. So this is a molecule that has been designed, again, based on a crystal structure, but it binds not exactly the same site as the estrogen binds, slightly in a different region. So this one also will be selective for the alpha receptor. And these two will be more selective for the beta receptor. And means selective, that means that if you measure what's called the binding affinity, this is the ability of the molecule to interact with the receptor. And if you do this for this pair versus alpha and beta and this pair versus alpha and beta, you will find out that one will be binding with higher affinity to one rather than the others. And that difference could be only tenfold, but it gives you already therapeutic window to work with if you want to modulate activity of one receptor versus another. Okay, now what happens once you have the coactivator recruited? First of all, I remember, so you've been asking, so what does coactivator do? Do they actually interact with other proteins? Or they, by all means, interact? Both of these proteins, P300 and so-called Crab binding proteins, are general coactivators that are being recruited by activated estrogen receptor. So once estrogen receptor is bound with estrogen, forms homodimer, and is on DNA, then this particular protein, one or the other, will come in contact with the activator function of the estrogen receptor. These proteins are actually quite big. So they are 300 kilodalton in weight. So they have thousands of amino acids and about 8 different domains. Both of them are similar in their function with regard to when they are recruited by the hormone receptors, but they are slightly different in sequence. So they are oftentimes called paralogs. And both of them have activity of histone deacetylase. In addition, they bind these so-called associated factors. So it's essentially what's being recruited, not only this protein coactivator, but some other proteins with it. And then those additional proteins will facilitate the recruitment of the other transcriptional machinery components. And so some of them will be this transcription factor 2D, which comprised of so-called TATA binding protein. And why it's TATA binding protein? Because it binds to the region of DNA in a promoter of the gene that has sequence TATA. And so the TATA binding protein then will be basically recruiting other co-regulatory factors that will then cause assembly of the RNA polymerase to holoenzyme. And then once this happens, you get another process that will activate RNA polymerase to holoenzyme, and you get gene expression started. Now, and interestingly enough, estrogen receptor, when it's activated, will be binding to this region, which has 2 leucines, and then there are 2 other amino acids irrelevant for that binding, but it's essentially a leucine-rich region in the N-terminal domain of P300 and CBP. So that's the recruitment that happens, and that's the example. So this portion is from the estrogen receptor, and that's the leucine-rich region, N-terminal domain of P300 and CBP coactivators. So very, very small portion of the protein responsible for its recruitment and binding to the activated estrogen receptor. But once this happens, then histone acetyltransferase function, HAT function of P300 or CBP will be turned on, and you get the chromatin remodeling. You get activation of gene expression. Okay, now, if you want to go into more details about how coactivators look like, so here they are. So P300 and CBP, very large, have a lot of different domains. This is the only region that interacts with estrogen receptors and other nuclear hormone receptors. That's why it's called the hormone receptor binding site or receptor interaction domain, like it is in here, abbreviated, and so on. Histone acetyltransferase domain is right in the middle of this protein. And then here is a binding site for this another protein, which is called P300 CBP associated factor, PCAF. What it does, it's responsible then for binding with other parts of the transcriptional machinery. And then there are other domains that are interacting with other proteins, which is not that important. There is this KIX domain that is interacting with the Crab protein. There are these zinc finger domains, which are not the same as zinc fingers that are in estrogen receptor DNA binding domain. So P300 has its own zinc finger type sites. And so actually there's a lot of structural data and structural information about this because these proteins really act as a coactivators for many transcription factors. For example, all nuclear hormone receptors that are in that particular table will be interacting with that region and then activating gene expression. There will be other transcription factors that have nothing to do with nuclear hormone signaling that may be binding to some of these domains. So these proteins are really like a Swiss Army knives of gene expression because they sort of be used for everything. And so they are very important in activation of gene expression. So there is also their smaller brother or younger brother, P160, so about half a size. And it has also nuclear receptor hormone binding site. And now what it does, it can do histone acetyltransferase action, but not directly, but essentially what it does, it binds first hormones and then could recruit on its own CBP or P300, and it has a binding site right here for them. So basically it's either P160 plus one of these, or one of these directly that will be serving as a chromatin remodeling machinery to acetylate histones.
And so does the receptor interaction domain bind to the N-terminal or the estrogen?
So, okay, so this you mean. This will be binding to the basically the activator function of the estrogen receptor. Not the same site as the ligand binds. It's a different one.
So is the N-terminal?
Yeah, activator function. Yeah. P160 basically binds to the activator function of estrogen receptor as well, just like P300 or CBP, except that it binds right in the middle of its sequence instead of N-terminus of this protein. But it will be binding to the activator function, same activator function as P300 and CBP binds. Okay. Now, just to show you one more time how this all works. So you can see when you have histone deacetylase is bound to the inactivated receptor, and that's not just the estrogen receptor, I meant that for any receptor that forms heterodimer that is already in the nucleus. Estrogen receptor is a little bit different in this regard. But once you have histone deacetylase or co-repressor bound, so that's the co-repressor bound. So histones are deacetylated, DNA is compacted, and it's tightly wound, you can see on this histone octamer. So 2 turns, 143 base pairs from here to there. 2 full turns, and then there is another histone octamer, another 2 full turns, and it continues like that, for billions of base pairs. Okay, and now once you have coactivator coming in, P300 or CBP, now histone tails become acetylated because it has activity of the histone acetyltransferase. And immediately, association between histone tails and DNA loosens. And the reason for this is very simple, because lysines on the histones have positive charge. And so when you don't have acetylation, there is a nice contact between positively charged lysines and phosphodiester backbone of DNA. So you form very strong electrostatic interaction. It's almost like, you have two charges interact with each other very closely. Now, in this case, you acetylated lysine, so there is no more positive charge. And so everything kind of slides out. And basically, the association binding between histones and DNA becomes much weaker. And so then thermal motion of the chromosome and all of the Brownian motion that may be in the nucleus will loosen it up and force it to be open. Okay, so this is the way it works. And it doesn't have to be extremely large gap between these particles or between these two coils to recruit more proteins, because what it does, once a protein binds right here in the middle, so this whole thing could slide. And so it's like you have a spool, between two strings, or two, you have a coil and spool, and so it can basically move back and forth. And so it's very, very easy then to open up the requisite amount, requisite portion of DNA for binding. So biologists came up with a fancy term for this, positional effect variegation, but it doesn't matter. So it's just, opening of the chromatin. All right, now, what kind of genes have been activated in response to the estrogen receptor activation? Well, estrogen response elements are actually present in hundreds of different genes. And in fact, you can identify them by doing the experiment which is called gene expression profiling. So what you need to do for this experiment, you take, let's say, cells population or dish with the cells which are coming from patient which has healthy cells, and you can have cells, second population, which is diseased cells or something. It could be, let's say, normal breast epithelial cells and breast cancer cells. And then in both cases, you add estrogen to the medium. And then after some time, you collect total messenger RNA of the cells and ask question, how many genes are being activated in response to estrogen in one case versus the other? And that's what gene expression profiling does that. So you have several conditions set up, or maybe hundreds of different conditions if it's hundreds of patients. And then you ask how many genes have been activated and what do they have in common? What these genes actually responsible for, what their protein products do. In some cases, it could be cells, it would be genes that stimulate growth of the epithelial cells. It could be genes that cause, for example, maturation of the embryo, or so on. And so by correctly grouping them, then you know what effect estrogen, for example, adding estrogen has on normal tissue and on disease state. And then you can go back and ask, okay, maybe these genes are under control of one of the isoform or the other. So which isoform of the two estrogen receptors will be responsible for activation of gene expression in diseased tissue versus healthy tissue? And then going back from that, you can say, okay, so if I know what molecule I could use to block particular isoform, and I will pick one that it will be for the disease state, I might be able to reverse that disease phenotype, or at least affect it for the patient. And that's what gene expression profiling does. So this is just the example. You don't have to memorize this whole thing. But so this tables are called, maps are called the heat maps. So they are showing you changes in fold of expression upon a different condition of treatment for the groups of hundreds of genes or thousands of genes. And the profiling takes care of the entire genome. Basically, every gene that is annotated right now and in a database could be interrogated this way. So if you look at, if you want to see all 28,000, 30,000 genes that you think are activated or changing in expression in the particular case, you can do that. Okay, now, so I just told you the way how one could use this strategy to basically design or use selective estrogen receptor modulators in different disease states. And so to simplify a little bit work of pathologists and also pharmacology work, different range of activity have been separated into different subclasses of these modulators. So for example, there will be molecules that will activate alpha and beta isoform in all tissues in every case, and estrogen is one of them. So whenever you add estrogen, regardless of how much you have alpha and beta isoform of the receptor, regardless pretty much of the tissue, it's going to go and activate whatever gene expression will be under control of this isoforms. Then you can have situation when it could be basically activated gene expression in one tissue, but repressed in the others. And so tamoxifen is one of them. And so apparently tamoxifen is quite effective if you use it on breast cancer cells. So whenever you have some phenotype that is already lost cell cycle checkpoints and so on, but it may be antagonist, or it could be agonist, sorry, in a tissue like, for example, ovaries or other organs, where you don't want to shut down gene expression and the estrogen receptor control. And you can have also antagonists that will be in all tissues, regardless of what type of isoform you're looking at. And so that's another example. Fulvestrant is one of them. And so the way how it works, first of all, different cells and different tissues may have, of course, different intrinsic expression levels of the receptors themselves, isoforms, alpha and beta. They may also have a different ratio of the co-activator or co-repressor proteins present. And also sometimes, depending upon molecule you use, even in the same tissue, you may be able to exert different effect on alpha versus beta isoform. And so that's the mechanism upon which it is based. And this is just the example of the selective estrogen receptor modulators. I am sure you probably already heard about many of them, so, and this is just, for you to refresh your memory, and you probably don't even have to memorize it because you already probably did or maybe will. There will be clinical, I think, people who will do this 10,000 times better than I do. So, but I just want to make sure that you know that there are this type of selective estrogen receptor modulators, which are used again in this particular conditions. Yeah. I would say probably okay. Because, if you say tamoxifen and it's used in breast cancer, I think, anybody you ask in the street, they will know it. That's so common, it's fine. And not so that I will be asking exactly what the mechanism of which is, but at least something you have to memorize, at least in this class. Okay, now, in addition, yeah?
Sorry, so is that a yes?
Okay, yes.
For like all of your tamoxifen?
Well, at least this part, okay? This part you may not worry, okay? All right. Okay, now, for estrogen receptor, remember I showed you this big scheme where you had many, many different pathways shown. So the reason for that scheme is that I wanted also to emphasize that in addition to acting as transcription factor, which is classical role, estrogen receptors can also do a few other things in cell. So first of all, I told you that most of the time they don't, and generally hormone receptors do not bind to the membrane. And this still holds true. But they oftentimes associate with the proteins which are bound themselves in the membrane. Like, for example, caveolin-1. So it's not strictly cell membrane, but it's little organelles which are called caveoli. So, and the caveolin-1 is one of the structural proteins in the caveoli, and apparently estrogen receptor binds to it. It is not clear what its function, and I don't really want you to, worry about this too much in this case. Only the reason why I told you this, that is that you can say, yes, in addition to acting as transcription factors, estrogen receptor can also have some other functions which are being studied and may not even necessarily be fully elucidated. So now also the G proteins could form complexes with the estrogen receptor, and some of this non-receptor tyrosine kinase is what I call it. So remember the map that I showed you with the phosphorylation sites of the estrogen receptors, and you saw this AKT, SARC, and so on. So some of these kinases could bind to the estrogen receptor and change its activity. So again, so just, as additional information. Then again, so now, through its downstream effect, through the activation of downstream genes, estrogen receptor also increases flux of calcium into the cells, and also increases levels of nitric oxide, although again, it's not directly, it's not doing anything to nitric oxide directly. It's the inducible nitric oxide synthase that is often activated by the estrogen receptor. And inducible nitric oxide synthase produces nitric oxide, which is a messenger molecule, and is used for different things in a different normal and disease state. And then also the activation of the estrogen receptor in certain situation where you have phosphorylation sites. So then you can regulate that by this enzyme, which is called the glycogen synthase kinase 3 beta. What it does, it is blocking phosphorylation one of the serines of the nuclearly localized estrogen receptor alpha. Again, so just for your information, so you can say that it's not just the activity of the hormone that may change the levels of expression of genes under estrogen receptor control. So some additional post-translational modifications like phosphorylation will still be able to tune up its activity, although main switch happens when the estrogen or selective modulators are bound. Okay, let's now talk about androgen receptors. So we don't actually have too much time. Whoa, actually we have only a few minutes. Fine. Now, so just like estrogen receptor, it is belonging to the steroid hormone receptors. It is called also in a subfamily tree in that big table that I showed you. It still regulates gene expression, and it's critical for the development and maintenance of the male phenotype, and it's very important in embryonic development as well. So this is then in embryonic development, it's regardless whether it's male or female embryo, it is required for number of different functions to be set up properly. Okay, so now this is the hormones that bind. So testosterone is one of them, and then through the action of the testosterone reductase, this dihydrotestosterone, or DHT, is being produced inside the cells, and so either this or the other one will bind to this ligand binding domain of the androgen receptor. And androgen receptor ligand binding domain has the most characteristic shape. You can see this several triangular alpha helices kind of connected to each other, and this is where the DHT binding site is. So very, very, very easy to recognize. Okay, now, just like in case of androgen receptors, you have two androgen, just like in case of estrogen receptor, you have two isoforms. Again, they are different in length. Again, they are somewhat similar in the activated gene expression. They have same classical structure as every other nuclear hormone receptor. So we have AB region and terminal regulatory, DNA hinge, ligand binding domain, activator function, C-terminal domain. Sometimes they are called ARA and ARB, so this is almost like alpha and beta, except for some reason biologists decided now to be slightly different in nomenclature. Okay, now signaling again is quite complex, but what we need to worry about is only this portion, as I said earlier. Oops, okay. Now, once you get hormone coming in, androgen receptor resides in the cytoplasm. You can see it bound by inhibitory proteins, HSP70, 90, and so on. So basically, so these four inhibitory proteins have been displaced. Now you have the hormone bound. There are a few other things happening there, so there are additional changes in conformation take place, and so it enters nucleus, and then dimerizes, and is a homodimer again, bound to the androgen response element. And once it's bound to the androgen response element, it's going to be recruiting P300 and CBP coactivators and activate gene expression. So very, very similar situation to estrogen receptor, except slightly different inhibitory proteins. And of course, the downstream genes that are turned on totally different from estrogen receptor. So this is another example how this looks like, but it's essentially reiteration of that signaling scheme. So this one probably a little bit easier to memorize or to follow. So you have testosterone, 5-alpha reductase enzyme makes DHT, DHT binds, heat shock proteins release. Now it's in a nucleus. It does get phosphorylated most of the time, not always, but most of the time. And then as a homodimer binds to its response element, recruiting P300 or CBP, and gets RNA polymerase complex bound, and gene expression happens. Okay, so what it does, basically. Well, you know, if people say something on steroids, I think they mean androgen receptor, right? Because what it does is essentially the anabolic effect to the body. And so it's muscle mass and strength and whatnot. Maturation of the sex organs and voice and whatnot, again, depending upon the level of androgen receptor. And in fact, even in the, during the embryo development, levels of the activity of the androgen receptor are very, very important. And sometimes aberrant expression of genes under AR control will cause problems in the embryo once it's born. Okay, so and then attention, memory, cognitive ability, again, so this is because of the general anabolic effect. Aggression, that's actually a very interesting part of the activity of the androgen receptor because one of its downstream genes is monoamine oxidase, A. And monoamine oxidase is responsible for degradation of the neurotransmitters and dietary amines such as serotonin and others. So essentially sensitivity of the brain to the different neurotransmitters will be modulated through the activity of the monoamine oxidase. And as a result, monoamine oxidase is regulated by the androgen receptor. So oftentimes they think that there is a link between activity of the androgen receptor and aggression, and also autism has also something to do with aberrant activity of the androgen receptor, although it's not quite yet been settled by people in the neuroscience field. Okay, now, progesterone receptor. One more thing, and we'll be done. Again, it's the homodimeric receptor. Interestingly enough, unlike estrogen or androgen receptors, it is being encoded by the single progesterone receptor gene. But now, two isoforms are still formed. Now they are formed through the differential splicing. So they are slightly different in molecular weight. So what progesterone receptor does, it basically contributes to the epithelial cell proliferation and also for the gestation of the embryo and during the growth of the embryo and so on. Interestingly enough, that progesterone itself, progesterone receptor, is a downstream gene which is regulated by the estrogen receptor. So activation of the estrogen receptor is important because it upregulates level of expression of the PGR gene itself. And then you can have a second level of control when you actually have now progesterone receptor activation. And so that's progesterone. Again, it's a steroid hormone. Somewhat similar by structure to estrogen and dihydrotestosterone, except it has acetyl group here, slightly different this ring, and so on. Okay, so this is actually, again, activator ligand for the progesterone receptor, and it is responsible for the upregulation of the gene expression for the genes that support embryogenesis and things like bone development, skin elasticity, nerve tissue, respiration, and whatnot. Okay, so the way how it works, well, so once it is in the nucleus, binding of the progesterone itself is actually changing the conformation of the so-called beta upstream segment of the progesterone receptor. So it dimerizes, homodimerizes, goes into the nucleus, and then conformational change causes it to recruit the co-activator proteins. And that's the way how gene expression program is turned on. Okay, so let's do just one more thing. Okay, and that's it. But you know, in return, so whatever we did not cover, you don't have to worry about an exam. So just what, because I gave you everything about the homodimer forming receptors. What about heterodimer receptors? So thyroid hormone receptor is one of them. It forms a heterodimer, and in fact, that's the way it works. So it forms the heterodimer with retinoic acid receptor, and it's been activated. So this heterodimer resides in the nucleus. And so when T3 or triiodothyronine is being added to the cell or enters the nucleus, it gets activated and then recruits P300 and CBP coactivators, which just like estrogen, androgen, or progesterone receptor. So these two hormones are actually produced by thyroid, and so thyroxine is then deiodinated to the triiodothyronine, and that's the main acting small molecule. Okay, that's it. So up to this point, and then if you have any questions, let me know. All right? Thanks. Yeah, we started a little bit late. We started 5 minutes after 10, right? So that's why. Or even. Okay. All right. Thanks.